We're diving into the fascinating world of immunohistochemistry with a focus on the synaptophysin test. If you're a medical professional or just curious about how we diagnose and study certain tumors, you're in the right place. Let's explore what the synaptophysin test is all about, the diseases it's associated with, and what you need to know before getting this test done. Presynaptic. Before undergoing a synaptophysin test, there are a few important pieces of information your healthcare provider will need. First, it's crucial to provide a copy of the histopathology report. This report gives detailed information about the tissue sample and any abnormalities detected. Second, the site of the biopsy is required. This tells us where in the body the tissue sample was taken from, which can affect the interpretation of the test results. Lastly, your clinical history is essential. This includes any symptoms, medical history, and previous diagnosis that could provide context for the test and its findings. Specification of the test. So, what exactly is the synaptophysin test? Synaptophysin is an integral membrane glycoprotein found on presynaptic vesicles in neurons. This protein is a key immunohistochemical marker, especially when we suspect tumors with neuroendocrine or neuronal differentiation. In simpler terms, if we're looking for tumors that have nerve cell characteristics, synaptophysin is a great marker to use. This test is particularly useful in diagnosing a range of tumors. Some of the diseases associated with synaptophysin include Gangliocytoma, Ganglioglioma, Neurocytoma, primitive neuroectodermal tumors, PNETs. These are all types of tumors that either originate from or affect nerve cells, which is why synaptophysin is so important in their diagnosis. Specimen used. The specimen used for a synaptophysin test is typically a biopsy sample from the suspected tumor site. The tissue is carefully prepared on a slide and then subjected to the immunohistochemical staining process. This allows the pathologist to identify the presence and distribution of synaptophysin in the tissue. Components include. The test includes several key components. Tissue preparation. The biopsy sample is fixed, usually in formalin, and then embedded in paraffin to preserve its structure. Antibody staining. Specific antibodies that bind to synaptophysin are applied to the tissue sample. Microscopic examination. The stained tissue is examined under a microscope to identify synaptophysin presence, which appears as a distinct staining pattern. Test report time. The test report time for the synaptophysin test is typically around 7 days. This time frame allows the laboratory to carefully prepare the specimen, perform the staining process, and thoroughly analyze the results. Your healthcare provider will then review these results with you to discuss any findings and potential next steps. In summary, synaptophysin is a crucial marker in immunohistochemistry for detecting tumors with neuroendocrine or neuronal differentiation. It helps pathologists accurately diagnose conditions such as gangliocytoma, ganglioglioma, neurocytoma, and primitive neuroectodermal tumors. Understanding how this test works and its significance can help in navigating the diagnostic process.